All right, why don't we go ahead and get started then? Chris, Yuri's connection is a little weak, so I was going to get things kicked off here for you. Thank you. First of all, thank you very much for joining us. I'm not sure how much kind of background you got, but here you have a DIY community. We have a once every 2 weeks journal club that we go through. And this is actually our first time having a PI join us. So we're very thankful for your time. We're playing a little bit with what the format might be. Initially, kind of the contact was made around a specific paper, but I think the consensus was a more broad, general discussion about synthetic bio and your interests, and kind of spread out from there, maybe with some directed questions about the paper, but have it a fairly broad and general discussion would probably be the most productive. So yeah. If that works for you?
Yeah, that sounds good. So, I mean, actually, the paper that you guys brought up is, it came out maybe a year ago, but it's actually work from about 5 years ago. So it's actually quite different from what I do today in my lab. But I'm happy to talk about the paper and talk about what's going on now and where things are headed.
Yeah, that'd be great. So maybe 5 years. So maybe kind of just a quick summary. How's it moved forward since then? I guess probably the more current activity is more interesting.
Yeah. So is that where we want to start? Want to start with talking about what's going on right now in my lab?
Right. Does that work for everybody?
Okay.
Sounds great.
Yeah. So basically, my lab at the beginning had two project areas. This tumor-killing bacterium sort of stuff that was kind of inherited from my postdoc. And then what I had started getting into was foundational technologies. And largely that was a reflection of kind of the ideas that were emerging around UC Berkeley that led to the development of SynBerk, and a lot of activities in labs around here that was kind of recognizing the fact that the big problem in genetic engineering was the tools haven't changed in 30 or 40 years. And while we wanted to keep challenging what we could do with genetic engineering and going into these new areas like therapeutic bacteria, it was also clear that we need to have a very focused effort on the foundational stuff. And, once we got through the paper that you're seeing, we kind of decided that the focus really needed to be on the foundational stuff more than the kind of the out there applications. That even with the applications that are the familiar ones associated with small molecule synthesis and production of proteins, that the way we're thinking about those systems is still overly mystified to a point where it's hard to move forward and build more complex things when we still haven't worked out how to correctly think about the simpler systems. So what my lab does these days is kind of centered around this thing called ACT Synthesizer. And what ACT does is it aggregates biochemical data and then uses chemical theory-based abstractions to bin the data into functionally similar groups. And then we have synthesis tools, where a synthesis algorithm is a piece of software that automatically finds solutions to problems. And in this case, the problem is whether a given compound can be made or not. Or more specifically, what this tool does is it enumerates all the chemicals that can be biosynthesized chemically. So we're able to do that. We pull in data from basically every source that exists, and we're able to compute every chemical that can possibly be made from that data, as well as any chemical that could be made if you were to speculate the existence of one additional reaction.
Okay, so can I ask a question here, Chris? When you say any chemical that can be synthesized, you talking direct chemical synthesis or synthesized through known biosynthesis pathways?
Biochemically. So, and we specifically mean things that can be totally biosynthesized, meaning you can just take the cell, put these genes in it, and it will produce this chemical product. So there are other scenarios where you would say use synthetic organic chemistry to produce a small molecule intermediate that you feed to a cell and then undergoes a biotransformation that converts it to some product. And that is not a scenario we currently deal with. And we also do not deal with anything that is chemical synthesis related, though we plan on doing hybrid synthesis and hybrid chemical and biological synthesis predictions as well in the future. So that's ACT. And actually, we just started a company around ACT called 20N. So in probably 6 months, that's going to graduate from my lab altogether, but it's still kind of the framing idea of the lab that we should formalize the process of design. And the reason for doing it is a lot of projects that kind of never made it into publication failed in the lab over these things that really should have been caught. And right now we're relying on, just the exceptional biochemical knowledge of individual practitioners to catch every design error because we have no software that can do this for us. And what we realized is when you just get these systems just a little bit bigger, like you start putting in 10 genes instead of 5, you very quickly run into the scenario where at least one of those genes is likely to do something in the cell that you weren't aware of. And it's not necessarily the case that it's not known to human beings or even to the public domain what those interactions are. It just might not be known to the individual doing the experiment. And so it just didn't make sense to me to be proceeding with trying to build these big systems until we resolve that issue. So that's the main reason we're doing this ACT thing, is to get that sort of formalism in place so that we can ultimately do verification of designs where we can predict what all those other interactions will be and rule out particular designs or suggest genome knockouts or other strategies to get around these problems. So that's a computational project. We also have another computational project called Clotho.
Yeah, I was going to ask a quick question on the ACT. Is that something that's a web service? Is it something that's out there?
Yeah.
Or is it private to the lab?
It's pathway.berkeley.edu. I think it might be broken right this second.
Okay.
Right now, so we're starting a company based on it, but there will be an aspect of this that is totally public. So it's not going to be the sort of company that kind of hides in the shadows. It's going to have a front-facing domain with the intent that it's not just business-to-business transactions, that anyone could interact with this data for non-commercial use. And if it's for commercial use, then there's some means by which that is done.
Great.
But yeah, for right now, what exists of it is online, but as of today, it is broken. If you looked at it 2 days from now, it would probably be fine.
So, in 2012, we were at a meeting that the FBI put together in California. That was DIY groups and other people. And I remember somebody presenting a program. It might have been someone from your lab that sounded very similar, where you could tell it what the starting molecule was and what the ending molecule was, and it would fish around and find naturally occurring enzymes that would convert through some sort of a pathway. In other words, it would find a path from A to Z.
Yeah, that might have been me, actually.
Yeah, because I remember this, and I remember there was so much excitement around it. It's great that it's gotten to this point.
Yeah, and there's some other people in this space. I mean, we're not the first people to try to do retrobiosynthesis. There's they're basically kind of two approaches. There is abstract synthesis, where you can, but you don't have any actual data fed into it, so it can never concretize anything. And then there's this other approach where everything is very concrete, where you're just tracing together. We mainly figured out how to do both simultaneously. So we were able to both say, these are all the pathways that absolute confidence that as well as the scenario where we say, well, this enzyme might work, but it also might not. So, a pathway that involves one of those predictions is you'd have less confidence over it than one that was one of the concrete ones. But that's the main novelty right now of what we do with ACT that is different from what's been done before. The next phase of it, though, is also inputting expression in stress and load data about the enzymes. So it's kind of the next shell of considerations that you would bring into the design experiment that allows you to say, well, even though that pathway maybe worked in its organism and it did all the right chemistry, we have data or predictions that say that that enzyme will not express in the cell you want to put it in, so don't use that pathway, use this other pathway instead. So kind of related to ACT is kind of what we do, what we're kind of refocusing the lab on now, which is multiplex characterization. And basically the idea here is figuring out how to get massive amounts of data using the new tools of multiplex oligosynthesis and multiplex sequencing. So basically deep sequencing readouts applied to genes that have been synthesized on these OLS microarrays, these things that Agilent is putting out and a few other companies where you print like 55,000 oligos on a chip. So they're protocols by which you can take these chip oligos and assemble them into genes. And we figured out a way of doing that as a library. So you never have to split it into hundreds of reactions. You can just make the genes assemble in one pot. And so what you get out of that is this ortholog library where maybe you synthesize 100 fairly similar, say, glycosyl transferases. And what you're trying to do is find from those libraries the substrates for all those enzymes. And if you can get this all to work, then the data gets plugged into ACT, which directly feeds into new predictions. So the way this works right now is the one way of working, we call it a cofactor depletion assay, where basically you take each gene and you put it into in vitro transcription translation mix in a tiny droplet. And right now we're doing this on microliter scale, but the intention is to do it at nanoliter scale or in emulsions or in droplets in microfluidic devices. And that makes the protein express, so you get a very tiny amount of protein. And then you spritz in a little bit of the substrate as well as a cofactor that is required for the reaction. And if that reaction takes place, then the cofactor gets consumed. And if the reaction doesn't take place, then the cofactor stays just where it was. So then in the second step, you spritz in a DNA probe that has some fluorophores on it, as well as an enzyme that will use that cofactor to modify the DNA. So if the first reaction had occurred, then the cofactor is gone, and there's no cofactor to modify the DNA. However, if the reaction didn't occur, then you still have cofactor, and the DNA gets modified. So the effect of this is you've recorded the outcome of the first reaction as a chemical modification on the DNA. And that DNA modification can be read out as a fluorogenic signal, which is what we have working now, but it could also be read out by deep sequencing. So basically you can, like let's say the modification which in this case is glucosylation of the DNA. If you glucosylate the DNA, you can block a restriction site. So if it's not modified, then the restriction enzyme cuts, generating a sticky end, and then you can ligate on adapters to that population. And then that population can be separated out for deep sequencing. So if you trace through that whole thing on paper as to the outcome, it basically comes out as if the first reaction took place, the adapters get ligated on, and you get increased counts of that gene in the deep sequencing. So the whole goal of this is basically figuring. Lost
I said,
you?
it's so elegant. That's awesome.
Yeah. Yeah, I never know if it's like people are lost by it or.
No. I'm
Yeah,
amazed
right?
the translational machinery would work at a nanoscale like that in like such a tiny.
Yeah, I mean, you're basically getting down to the size scale of a real cell in these droplets. Yeah, so it's, you still have probably hundreds of ribosomes, but not millions. But you definitely can scale this stuff to that scale. Yeah. So we know we can do this assay for N80 plus, UDP glucose, and S-adenosyl methionine. And that may actually be all one needs to watch the rest of biochemistry, simply because those reactions are incredibly diverse. So pretty much every chemical should have some enzyme that does at least one of those three reactions, because they're promiscuous glycosyl transferases and promiscuous methyltransferases. And redox chemistry is it's by far the largest class of reactions that exist. So the thinking is we could actually use these as biosensors ultimately, where you pair up the enzyme that can modify the product of your reaction sequence. And if your reaction sequence works, then you make the product and you get a signal in the deep sequencing. And if your reaction didn't work, then you don't get a signal. So you're using basically an enzyme as a biosensor in this scheme.
You say you can get a signal or not get a signal, and it sounds binary, but you can actually quantify the level of signal with that. It sounds like you should be able to.
Dynamic range, but you definitely get a graded signal. I mean, what you get out when you do this assay is, some things do absolutely nothing, and some things fully consume the cofactor, but other things fall somewhere in between. It's just not a huge dynamic range, but that actually is not limiting. It does seem to be fairly quantitative, though, at least when we do it at this sort of, several microliter scale. Like run to run, even when you get a partial signal, it's a very similar partial signal, at least plus or minus 10 to 20%.
Hey, Chris, can I inject real quick? I forgot to kind of do any introductions here. I was thinking maybe it'd be useful for you to just be able to pin a name to the voices, and maybe just as people have questions, they can introduce themselves. So my name's Roger Steger. Derek and Ellen, do you want to introduce yourself?
Sure, I'm Derek Jacoby. I have some involvement with BioCurious down in the Bay Area, but also run a space of my own, BioSpace up in Victoria, British Columbia.
Oh, great.
I'm Ellen Jorgensen. I'm a co-founder of GenSpace in New York. I'm also an adjunct at New York Medical College, and I've been in the biotech industry for a while.
We met at SB6 briefly.
What?
I think we met at SB6 briefly.
Oh, yeah, we did. I've got such a sieve for people's faces. And it's awful. I'm sorry. I think we did. Was I sober?
I think so. I wouldn't know, but I think so.
Keith, you're on.
I'm Keith Camito. I'm a computer science and mathematician person, and I'm a member of GenSpace. So I'm recently transitioning into the biospace and having a good time of it.
Great, great.
And I'm Oliver, another co-founder of GenSpace. I am zooming in from Cooper Union, who's finishing up a laboratory here for a course that I'm teaching, and students there are sitting behind me to take off because the class has ended and it's getting late, so I'm sticking around.
Okay.
I'm Daniel Grushkin, another co-founder of GenSpace, also a fellow here at the Woodrow Wilson Center, working on a book about synthetic biology, and right now in Washington, D.C. for the next couple of months.
Cool. Great.
I'm Dave Casey. I'm based in Florida. I'm a member of BioCurious, and I'm trying to get a space started down here in Florida.
So I didn't mean to interrupt the flow, Chris, if you wanted to keep going on. But you can see we have a nice, I think the name of the group, Google group is Coast to Coast, DIYbio Coast to Coast. We got Canada to Florida and Cupertino to New York, so pretty broad.
Terrific. Yeah. So you have physical space with, most of you are from the New York space, right? The, I forgot what you call it, but the gene space?
GenSpace.
Yeah. At least 4 of us are from there.
Yeah, yeah. And that is,
You're looking at it. It's right here in the space.
and where is it exactly? Is it like associated with other science stuff, or is it a standalone thing?
We always collaborate, but it's a standalone nonprofit organization. But we're always collaborating with one organization and another. Ellen actually set up a collaboration with Cold Spring Harbor, the DNA barcoding events that were taking place at GenSpace. But yep, we're a standalone entity.
And are you doing individual projects there? Is it like a workshop where people can come in and work on their own thing, or?
That's exactly the. So it's basically half and half. We have people working on projects there, and we also have people. So that's essentially how it was founded, was for people to work on their individual projects. But it's since then, we've been offering classes to the public, which I don't know if Ellen's in the middle of a class right now, but we've certainly had a lot of members from the public come in and take classes in genotyping and synthetic biology and crash course in biotech that Ellen set up. So we've been doing a lot of educational outreach as well.
Mm-hmm. Terrific.
Yeah, I'm running a workshop at Yale tomorrow that I'm preparing for. So.
Okay.
We're going to do the Genomicon thing. You know, that thing from the Alberta team? Alberta
I'm
IgG
really curious to see
data.
how that goes, Ellen. I've actually got one of their beta kits as well that I won't get to a class for until December. But yeah, that's really exciting stuff, isn't it?
Cool.
Chris, can you tell us a little bit about your trajectory, your career trajectory, how you got to where you are, and how you got to be studying what you're studying?
Yeah, sure. So when I was an undergrad, at the beginning I had no idea what I was wanting to do, but I was really good at chemistry. So I started in chemistry, and then it was my sophomore year, I kind of serendipitously joined a lab, and it turned out to be a polymer chemistry lab. And at the time, I didn't really get what chemistry was about as a field. It was just kind of on a whim that I joined a lab. But I kind of took to it very quickly and got really into it. So I was really interested in polymer chem and kind of process engineering at the beginning, and mainly that was because the lab that I joined was doing that. But as I kind of was advancing through classes, I got turned on to particularly protein engineering. So I had read a book, like a popular book, I forget what it's called, but it was, talking about crystal structures of proteins, and I was kind of blown away by it. I was just kind of amazed that that's how it all worked. And the further I got in the classes, it became clear that both my strength and my passion was around the biochemistry. And specifically what I wanted to get into was protein engineering. And actually, a lot of the stuff that I was interested in when I was an undergrad was this sort of metabolic engineering stuff. I was really interested in small molecule biosynthesis. And then I went to grad school, and I ended up going to Scripps Research Institute and working with Peter Schultz. I had originally been interested in doing like directed evolution stuff or like that RNA selection kind of stuff, like Jerry Joyce stuff. But then rotated doing those things and realized they were kind of all process and not much theory. And realized I really wanted to work on something that was more design oriented, that required some more science knowledge and less intuition. And so I joined Schultz Lab and was working on site-specific incorporation of unnatural amino acids. And the particular areas that I was put on originally for that project was core base codon stuff, and that all worked. And kind of closer to the end, I got more interested in the problems that centered around either trying to biosynthesize unnatural amino acids before they're incorporated into protein, or deal with unnatural amino acids or amino alcohols that get degraded by the cell, and figuring out how you eliminate those degradation pathways. So we ended up doing the alpha hydroxy version of tyrosine and incorporating that into protein. And we also had a cell that biosynthesized para-aminophenylalanine and incorporated that into protein, all in the cell. And kind of through all this, like I feel like at the beginning of this, I had this perception that you really could only put one gene into a cell, and that was the scope of genetic engineering. And I think a lot of people were thinking that way. And it was experiments like the Schultz stuff where, some of these cells that we were creating had 7 or 8 genes in them. And it became clear that thinking that you can only put one gene in is just flat out wrong. I think nowadays we kind of have a more balanced view of that statement, because I think a lot of us felt out just, okay, how far can you push this thing before things start breaking? And we have a much clearer notion as to what the issues are. But at the time it seemed like, oh, you can do anything to these cells. And so that got me into wanting to really push the limits of just how far can you push this approach of genetic engineering where you're adding genes. And it was about that time, that was like 2001-ish, that I was graduating or kind of finishing up in Schultz Lab. No, it was 2003 that it finished up. And, synthetic biology had been going for a year or two at that point. Things like the toggle switch and the repressilator had come out. And that seemed like the right direction to go. So I started looking around to who to work for, and ended up with Adam Arkin and Chris Voigt. And Voigt was actually in Arkin's lab at the time, and that's how it became a joint thing. So I ended up working in Chris Voigt's lab for, 2 years, and Arkin's lab for 2 years, and we were working on therapeutic bacteria. Got a few papers out on that. And the last one was that payload paper, which we did in my lab proper. Yeah, and then the transition from that to what I'm doing now is kind of just the recognition of, it just became really clear when we tried to attack this really big problem, just what exactly the problems were that needed to be solved in a way that was not clear going into that project. Like, we kind of dove into doing therapeutic bacteria not knowing what we were going to hit or what we're going to run up against. And it just became clear by kind of the third project within it just what the issues were. I mean, not necessarily issues specifically with that particular project, but the problems in general associated with synthetic biology, this idea of adding genes into a model organism to create new function. And we did enough projects to see what things work and what things break. So now I'm just really focused on the theory and the methodology to get this such that it's much more reliable, much more predictable, and you can kind of rule out one design over another that will work.
So Chris, you shifted away more to the foundation, away from kind of the therapeutic, kind of that level. Are you working with people that are kind of working on the applied side of it at all?
Well, so what I realized too is if you're approaching this from a foundational perspective, you need to find the frontier that you can see direct benefit of those tools on. And therapeutic bacteria is like the most advanced application you could possibly think to do with a bacterium. Like, you not only are having to master biosynthesis and transcriptional circuit kind of control to make that happen, you also have to master the knowledge of what is going on with immunochemistry. You're having to deal with doing all these predictions about quantitative expression levels in a foreign environment where you can't actually do an experiment, because we had gene expression changes happening within the cytoplasm of a mammalian cell. And that was a lot of the trick of that payload paper, was we're trying to make a program that turns on when it is inside a cell. So actually just figuring out okay, how do you even characterize a promoter when you can't just grow it in a flask, because that's not going to give you meaningful data. The phenotype only occurs when you put it inside a cell. So how do you do that in a quantitative way? I mean, it is the, like, there isn't something that is more complex that you would think to do with bacteria other than a therapeutic. And it became clear that, if we're really trying to do this all very rationally, methodically, and reliably, therapeutic bacteria is the last one you do, not the first one you do. So that's why we're focused on the small molecule stuff right now. But I don't view this as a long-term thing. This is probably something that I'll do for another 2 or 3 years. And then I want to get back into doing kind of immunochemistry. So one particular piece of the tumor-killing bacteria stuff that I think we can get much more traction on a 10-year time scale than we probably could have with such a complex system as a therapeutic bacterium. So this would be things like trying to identify either specific structures of carbohydrates or small molecules that kind of trigger the immune system to do things. So they're drug-like things in the sense like they're small molecule chemicals, where you're producing those chemicals in cells, and they don't inhibit receptors. Instead, they tell the immune system to do things. So I think that's probably where we're going.
So directing the immune system to treat the disease, is that kind of what you're...
It's kind of like a vaccine, but I haven't worked through any of this yet. But there's certain chemicals that bacteria will make, like cholesterol glucoside is one that Helicobacter makes. And they're basically just small molecules that directly communicate things to the immune system. It's more like, talking to the immune system more than eliciting adaptive responses or trying to inhibit some receptor. They're more like small molecule communications. And I think there's a lot of natural diversity that we can probably mine from in doing this. But I think we can also approach this as mapping out the chemistry of the immune system. Yeah, I mean, I still think that the therapeutic space probably is the big killer app area for synthetic biology. I think there's a nice area around materials, which is kind of what we're in right now with doing apps.
Just to follow up, and this is probably a general question. You know, I think we're all in this forum familiar with the grand goals of synthetic biology. How would you characterize the state of the art?
Just as where everything fairly.
And the second question I had was, you know, what are the next small steps in synthetic biology? What are the problems that people have to solve now to get us to the next level?
I mean, unquestionably, the number one is the gene synthesis costs and scalability and reliability. I mean, I think that is what drives everything. And the reality is, like, I feel like these sort of multiplex assays that we develop, you know, it's clear that these things exist. And yes, we do have to make these things and we have to show that they work. But I don't have any doubt that it is possible to make these things. And once the synthesis is cheap, it will be easier to make the rest of the assays. And certainly in terms of kind of turning the crank and just characterizing tons of genes to get the data that we need going into the databases, that can't really happen with the current prices of gene synthesis. Like when you just price out a project with the sort of budgeting that would be appropriate for, you know, granting agencies or investment from a company and the like, just the price points we're at right now kind of limit what you can realistically do as a mining effort. So like if I want to go make a specific chemical, I definitely can do a very thorough sort of mining effort around enzymes that might do a specific chemical. But if I want to do kind of just a general survey of the space and synthesize, you know, 100,000 genes that all are one clade of enzymes, that's just not realistic today. But actually the only limitations on getting data on that scale is the cost of the DNA synthesis. Like the sequencing is there, the assays are there, it's just the synthesis. So yeah, I mean, I think it kind of all boils down to that.
Kind of reminds me of the early days of microarrays. I remember when one microarray was like $20,000.
Yeah, and it'll come down. I mean, so, you know, there are a lot of entities working on this problem, obviously, and it seems to be largely a problem of figuring things more than actually fabricate them. Like many people are saying that the price per oligo, once you erase kind of all the added arbitrary cost to just make more money off of things, you actually, you know, consider the cost of the reagents and such associated with the microreagent synthesis, that actually the price point is pretty good. It's just the difficulties associated with making all those things reliably assemble and, you know, sequence confirming the product. Those are the bottlenecks. So those really should be solvable. You know, George Church had a paper where they linked together microarray-derived gene synthesis and deep sequencing readouts. And I know folks have thought that their particular strategy was probably not deployable yet. But, you know, so I say that's one example of how to do it. And there are probably other ways to do it. And it seems like there are a fair number of people working on that specific problem of how do you combine the deep sequencing with the multiplex fabrication, such that you get you're eliminating all the bottleneck steps associated with fabrication and doing that.
There's also a price point associated with anything high throughput. And what I'm kind of interested in is, as a member right now of the DIY community, I sometimes wonder, it seems like synthetic biology is high throughput.
It is now.
Wisdom about how the amateur community can contribute to this field when we don't have this sort of stuff hanging around?
So I think part of it is that will change when these become emulsion methods rather than high throughput screening methods, or they become microfluidic methods. And I definitely am approaching the assay development with the assumption that it actually only makes sense if you do it in an emulsion or a microarray. Like the volumes associated with bona fide high throughput screen, where you're saying using tips and liquid handling robots, that's actually that would be a very expensive way to run these assays relative to what you could do in an emulsion. And actually emulsion stuff, I think a droplet generator, I haven't gotten a quote yet on what they cost, but my guess is they're probably a fairly low cost sort of instrument, in the sense that it's just a tiny tube with a valve. It's not doing anything particularly fancy. So I think that will happen, that it actually will be possible to do high volume acquisition outside of the context of academia, but I wouldn't expect that in the next 5 years. I think, though, we can have assays that are these sort of all in vitro enzyme coupled sort of things that are a little bit expensive to run at the volumes that you can do with your hands. Like 10 microliter scale is basically a $10 reaction, but there's no equipment investment associated with those experiments. And if you to just clone out a gene from a natural source, you're already over the $10 or whatever associated with the assay. And if you gene synthesize today, you're looking at $200 for a gene. So that's obviously far in excess of the cost of the assay. So I think, you're not going to be able to do today hundreds of thousands of data points of characterization outside the context of a very well-funded effort. But, I think these tools actually do enable DIY stuff to happen on a smaller scale. So actually, studying particular genes that no one else is looking at, applying a few new observations that go into the public domain. That's a very valuable contribution. The other thing is, there are kind of two flows in my mind associated with the field. There's the characterization phase, where you're going and characterizing individual genes to generate electronic data. And there's a second phase where you use electronic data to design an organism that does something new and interesting. And all of these software tools being developed, they're all oriented towards being kind of openly available, open source things. So, ACT predictions, for example, those things, you're going to have access to at least a subset of the pathways, if not all of them, for non-commercial use. And the intention is to enable people to use these tools, give them kind of access to knowledge that is usually unavailable unless you're, a Jay Kiesling kind of person. Just like in terms of having that notion of like the lay of the land of the biosynthetic space, that kind of makes it such that you don't really have to know that much except the basic rules of biosynthesis, because it does the calculation for you of where to start in terms of what chemicals are going to have a solution and what the options are for the genes that you would use to make that chemical. So I think there's a fair amount of stuff being developed both in my lab and outside that actually are quite accessible to a DIY sort of effort.
A question about something that starts to branch into some of those DIY efforts. You've helped run the Clotho team for a number of years on the iGEM teams. This is the first year this year that community labs like ours can participate in iGEM. Do you have any thoughts on what that kind of model can do to interactions between community labs and academia?
So I think, I actually started talking to DIY folks in the Bay Area maybe 3-4 years ago, and I try to keep in touch. And I think realistically, it's still probably the case that the most effective place where someone outside of a conventional wet lab can interface with synthetic biology is around the software, simply because it doesn't cost anything and it doesn't require much technical training. You have to know how to code, obviously, but it's a lot easier to learn how to code than it is to learn how to clone. And yeah, so Clotho, actually, that is still an active area of research. I do that with Douglas Densmore. We're gonna schedule the release of the next version of it is next fall. From the 2010 release, which is the last release we had. So we actually got real money, real grant to develop Clotho. So we have full-time programmers, one on Douglas's end and one on my end, who are rebuilding Clotho as a web-based deployment. There's also the Autodesk thing called Cyborg, which is a similar sort of thing to Clotho in the sense that it's a platform and they're coming out too. And the intention with these projects is to create this kind of shareable dynamic content layer where you can create tools, create data, and share them very easily with other people. So I think there's a lot of opportunity for DIY effort in that area, more so in the lab side right now. But I think the wet lab side will also become more accessible.
I got a question about the coding of this. Is this going to be open source as well, or just the usage of it with the API? Or will people computer programmers like myself be able to see what's actually going on under the hood?
Clotho is open source fully, like a BSD license. No intention to change any of that. Cyborg is not open source. Content generated within Clotho, whatever by the developer of that. So the platform that is Clotho is open source, but some content within it.
So, Chris, we're coming up on an hour. I want to be respectful for your time. Is there maybe you have time for one more quick question? Anybody?
Earlier we were discussing, sorry, I muted my video. Earlier we were discussing, it is great that you were spending time with us. We're interested in attracting other IPs to spend some time with us. What would make this worth your and other IPs worthwhile to hang out with us for an hour? I don't know if that's articulated well, but that was what you're wondering.
I think you'll find both amongst academics and industry. You'll find that a subset of individuals are interested in engaging with the DIY community and others are not. And for those of us that are interested in what happens in the DIY community, I think we see it as an opportunity to just keep in touch. So it's not a hard sell. It's more just hitting the right people.
Well, thanks for hanging out with us. We appreciate it.
No problem.
And Chris, in terms of just the format, then does this work for you, or is there a different format? Do you like the open-ended? Do you prefer more directed questions?
Yeah, I think so. When I looked at the questions that I was sent, they were actually more specific than I would have been able to answer. Like questions about why was one cell line used in a figure or not. Those are questions that the student who did the study could answer. And actually, Jinha he's now in the Ron Weiss's lab. So actually, if you really want to know the real guts of a paper and the guts of experiment and the micro decisions about the experimental design, usually that's best to go straight to the first author person who actually did the experiment. There was probably a time when I knew those answers, but that was 5 years ago.
All right, great. Well, thank you very much for your time. We really appreciate it. Like I said, this is our first time. I found it very interesting, and I'm sure the rest of the crowd did as well.
Great. Yeah, I appreciate having the time to talk to you.
Thanks, Dr. Anderson.
Thanks a lot. That was great.
Thank you. Yeah,
Bye-bye.
thanks.
Good night, then.